I'm Dr. Amit Bhattacharya, Associate Professor from Department of Zoology, Ramjas College, and I'm speaking about the applications of the antibody-antigen interaction patterns for the development of the various immunology and the medical diagnostic tools. Now, in the first part of the lecture, we discussed about some of the important techniques like the radial immunodiffusion method, the double immunodiffusion method. So, taking that lecture further, we will be talking about the immunoelectrophoresis over here. Now, this immunoelectrophoresis method has been further utilized for the development of the various scanning or the screening of various antibodies present in the patient samples. Now, this immunoelectrophoresis it combines the electrophoresis and the double immunodiffusion method over there. Now, antigen preparation is first electrophoresed over there, and based on its charge component, they are separated over there. So, we can see over here the antigen is first loaded in the central well, and based on their charge, they move towards a positive or the negatively charged. So, if the sample is negatively charged, it will move towards a positive charge, and if the sample is positively charged, it will move towards the negative charge. Now, subsequently, on the top of the well, there is a trough which is there. So, the anti-serum or the antibody is loaded onto this one. Now, the trough can be on the either side or on one side of that particular well. So, once the antigen which are separated, the antibody or the anti-serum is loaded onto that particular trough, and it diffuses over there. So, the double diffusion now takes place. So, the antibody diffuses and the antigen we know have already diffused, and it has come to a position based on its charge. So, at that particular point, there is a line of precipitation which is formed over there. So, the precipitation line is formed where the specific antibody and the antigen interacts over there. Now, the immunoelectrophoresis has been used for the detection of the various or the absence of various proteins in this serum, majorly in many types of cancer, immunodeficiency diseases. It has been utilized. So, this technique have been pretty useful and very instrumental in determining whether a patient produces a certain abnormal amount of one or more antibody isotypes and characterize this certain immunodeficiency syndromes or the cancer diseases. Now, this particular image shows how exactly the immunoelectrophoresis is functioning. So, we see that the antigen is loaded into the central well, and as the antigen is loaded, the antigen moves or gets electrophoresed based on its charge. So, towards the positive or the negative charge, it moves accordingly. Now, once it has moved and it has diffused, what happens is the anti-serum or the antibody, it is loaded onto the troughs on one side of the separated antigens, and it is allowed to diffuse. So, over a period of time, what happens is the antibody also diffuses, the antigen also diffuses. So, there is a precipitin line which is formed, which is specific for the antibody and the antigen interaction over there. Now, this particular line then gives an idea that what amount and how much concentration of these particular antigens or the antibodies are present. Now, the electrophoresis phoretic pattern shown over here is present checked in the case of the myeloma cancers. So, in the myeloma cancer, it is being found that the patient produces a large amount of monoclonal IgG antibody. So, IgG antibody which contains the gamma light chain bearing antibodies which are present. So, a large number of monoclonal IgG antibodies are produced. So, the patient sample is taken up, and a sample of the serum from the patient is then electrophoresed. Then the anti-serum specific to the indicated antibody class was placed. So, the sample was loaded into the central well, it moved or diffused towards based on the charge density, it electrophoresed, and then the anti-serum for specific for that particular antibody class was placed on the top of the well, which was the trough over there. Now, over a period of time, the diffusion took place. So, at the concentration of the patient serum used and the anti-IgG and the anti-antibodies produce a line of precipitation over there. So as the interaction between the IgG and the anti-IgG takes place over there, there is a line which is present over there. So we can see the image over here. The IgG line is precipitin line is also formed. They checked for the IgM line also over there. Now, the agglutination reaction have been further utilized for various development of various other techniques also. One of the most interesting one is called as the blood grouping or the blood typing. So in this particular process, what happens is the interaction between the antigen and a particular antigen, it results in the visible clumping, which is called as the agglutination. Now the antibody which produces such kind of reactions are called as the agglutinins, and the agglutination reaction are similar to the principle based on the precipitation reaction, where there is a reaction taking place between the antigen and the antibody which is specific to a particular antigen or the epitope, and as the binding takes place, there is a precipitation which is formed or the agglutination is taking place. Now, one of the scientists in the year 1930s, he got the Nobel Prize, Karl Landsteiner, for the development of the blood grouping type. So this particular method became very instrumental in the subsequent years. Now what he did is he used this particular technique of the antigen-antibody interaction patterns only. Now we know that in the ABO blood grouping types there are various blood groups like A blood group, B blood group, AB blood group and O blood group. So depending upon the presence of the antigen, so in the A blood group what happens is there is a presence of antigen A which is there and anti-B antibodies which are present. Now in the B type blood group there is a presence of the antigen B and the antibody against the A antigen which is present. So anti-A antibodies are present over there. Then the type comes AB, where there is presence of both antigen A and B, but there is an absence of antibodies present in that particular individual. While in the case of the type O, there is neither A nor B antigen which is present, only the antibody anti-A and anti-B which are present. Now how exactly this particular technique was used for the screening of the blood grouping? Now they also use the similar kind of method which we just discussed, which is called as the agglutination. So what happens is whenever there is a specific kind of antigen, it will interact with its specific antibody, there will be agglutination taking place, reaction taking place, and it is visible in that particular solution. Now what we do in this particular to determine the presence of the recipient's RBCs, whether it contains the specific antigen or not, a blood prick is being done in the finger and the blood is taken up. Now the blood is taken up and there are various antibodies which are present, anti-A, anti-B and anti-D, which corresponds to the Rh factor, Rh positive or Rh negative. Now depending upon the presence of the antigen, the agglutination reaction takes place. So suppose there is a antigen A which is present in the blood RBCs, this will react with the anti-A antibody which is added to these particular samples, and there will be agglutination reaction over here. So we can see the image over here, the agglutination reaction is taking place over there. Now this particular reaction then gives us an idea what kind of blood group the person is having. So we can see over here in the type A and type AB kind of blood grouping. So as we know, there is presence of antigen A as well as antigen B type which is present. So there will be agglutination reaction both in A and B samples. So anti-A and anti-B, both samples will give the agglutination reaction. While in the case of the type A over here, we know that only antigen A is present. So only anti-A serum will give the agglutination reaction. The anti-B serum won't give the agglutination reaction because there is an absence of that particular antigen in that sample. In the type B, what happens is the anti-A serum won't give any agglutination reaction, but anti-B will give agglutination reaction because of the presence of the anti-B antigen B present in that particular sample. Now, in the presence type O blood group, what happens is there won't be any agglutination reaction either in anti-A or in anti-B reactions over there. So, there won't be any reactions taking place. So, how do we read our sample results over here? So, we can see over here, if it is O positive, which means that neither anti-A nor anti-B agglutination is present, but anti-D agglutination is present, which means that the RH, it is RH positive. So, this is RH positive reaction. So, it is O positive. In O negative, it will be negative for all the antiserum reaction, whether it's anti-A, anti-B or anti-D. In the case of A positive, there will be agglutination in anti-A only, but absent in anti-B, but it is present in anti-D because it is RH positive. In the case of the A negative, it will be only an antiserum A, which the reaction which takes place. Then B positive, it will be in the anti-B and anti-D. In the case of the B negative, it will be only present in anti-B over there. In the case of AB positive, it will be on anti-A and anti-B reaction as well as the RH, which is anti-D. In the case of AB negative, it will be anti-A and anti-B reactions over there, while the last one is the control or the invalid results which are shown over there. So, that's how we read our results in the agglutination reaction, and it became one of the instrumental techniques till now to check the blood grouping or the blood of that particular person. Now, the agglutination inhibition reaction or these particular techniques have been further utilized in the development of various kits. One of the important kits which have been developed over the years is the home pregnancy test kits which have been developed. So, we know that the female who is pregnant will be having a higher concentration of the HCG hormone, which is human chorionic gonadotrophin hormone. So, the HCG hormone concentration will be higher in this particular pregnant ladies. So, if we can test or check the sample for the presence of this HCG protein or the antigen with the help of the specific antibody, we can say that whether that particular sample is positive or negative over there. Now, this agglutination inhibition method have been utilized for the development of this particular kit over here. Now, how exactly this home pregnancy test kit functions in the thing? Now, what happens is there is a presence of an adjuvant which is there, which contains the antigens on the top of it, along with there is a anti-HCG antibody serum which is there. Now, what happens is first the urine sample is added with the antibody or the anti-HCG antibody over there. Then the hapten or the antigen sample is added over there. Now, what happens is if the urine is having the HCG concentration in a higher number, what will happen is it will bind to all these antibodies over here. So, the antibodies will be already utilized and they won't be able to bind to the subsequent hapten which is added into the sample. So, there won't be any agglutination reaction which is there. So, there is an inhibition of this agglutination reaction which will take place. But if the female is not pregnant, so there won't be any higher concentration of the HCG hormone in that particular sample. So, what happens is the antibodies, anti-HCG antibodies will be free as soon as the second hapten which contains the antigen is added. So, there is a mesh or a precipitation which is taking place, or the agglutination which is taking place, and the line will be shown over there. Now, this particular immunochromatographic technique was further used in the development of the dengue test kits also. So, this one-step dengue NS1 antigen test kit was also developed, and we know that what happens is in the dengue patients, the concentration of the NS1 antigen or the specific protein is higher. So if the person's NS1 antigen can be determined or checked, we can easily see that whether that person is suffering from dengue infection or not. Now this particular antigen-antibody interaction pattern was used for the development of the test kit over there. So it is called as the immunochromatographic assay over here. Now in this particular assay, what is being done is, first the serum sample, after the centrifugation, it is loaded onto that particular well. So what happens is, if the serum sample is having the NS1 antigen, it will diffuse over there, and subsequently the anti-serum is added onto the well. So if there is a presence of NS1 antigen which is present in the sample, so what will happen? The antigen-antibody interaction will take place, and there will be a line which will be there which gives a positive test over there line. So it can be easily made out whether the person is suffering from the dengue infection or not over there. So this particular antigen-antibody interaction was utilized to develop this particular medical diagnostic kits also. Now the next important methods which came into the picture was the radioimmunodiffusion assays. So in the radioimmunodiffusion method, if the name is telling you, what is being used is the radio labeled molecules are being used. So this is one of the most sensitive technique for determining the antigen or the antibody. So it is called as the RIA, or radioimmunoassay, which is there. This was first developed in the year 1960s by Barson and Yallo, and to determine the level of the insulin and the anti-insulin complexes in the diabetes. So we can see how exactly these antigen-antibody interaction patterns were utilized for the development to develop check the various kits with the help of the level of insulin and the anti-insulin complexes in the diabetes. Now subsequently, Yallo got a Nobel Prize for this particular breakthrough thing. Now this particular technique have been utilized to measure the hormones, serum proteins, drugs, vitamins at a very low concentration also. So it can be very useful in determining the amount or the measures of the hormones, serum proteins in the samples. Now this for this particular discovery in the year 1977, Yallo got a Nobel Prize, which he shared with the other Nobel laureates over there. Now in this particular technique, what is being done is the competitive binding of the radio labeled antigen and the unlabeled antigen to a highly specific affinity antibody is being determined. So we can see over here in the well, there is a presence of the antibody. So the preloaded antibodies are there, onto which the unlabeled antigens are being attached, and as this labeled antigens are being loaded onto them, there is a competitive binding. So these unlabeled are being replaced by the labeled one, and as this labeled one gets bound, we can determine the concentration of these labeled, and then we can check with the help of the standard curve to know the unknown concentration which is being added over there. So this method laid the foundation for the development of subsequently other methods such as the ELISA, enzyme-linked immunosorbent assays also. So as this concentration of the unlabeled antigen increases, what happens is more labeled antigens will be displaced from the binding site, and the decrease in the amount of the radiolabeled antigen bound to the specific antibody in the presence of the test sample is measured to in order to determine the amount of antigen present in this sample. So this method was used over there. Now this is the standard curve which is being plotted over here. So this plot is obtained by adding increasing concentration of unlabeled HBsAg, which is the surface antigen on the hepatitis B viruses, to a fixed quantity of the labeled HBsAg over there with these specific antibodies over there. Now the next technique which came into the picture was called as the enzyme-linked immunosorbent assay, commonly called as the ELISA. Now this was similar to the principle of the RIA technique, but it depends upon the enzyme rather than the radiolabeled method. because the radiolabeled probes can be difficult to handle because of their radiolabel properties. So they have to be disposed properly, else there will be a problem of radiolabel. So this particular method took into advantage because it used certain fluorescence probes which were called as the fluorochromes over there. Now, an enzyme which is conjugated onto the antibody, it reacts with the colored substrate to generate a colored reaction product, and such substrates are called as the chromogenic substrates over there. Now, a number of enzymes which were used in the case of the ELISA, which included the alkaline phosphatase, horseradish peroxidase, galactosidase, which was used, and this particular technique had certain advantage over the RIA because it was more safer and less costlier also. Now, the first technique which was used was called as the indirect ELISA, which is there. Now, the indirect ELISA, the method of the choice was to detect the presence of the serum antibody against the human immunodeficiency virus, which is the causative agent of the AIDS. Now, this particular method was used to check the presence of serum antibodies against specific conditions in the AIDS. So what happens is, when the person is infected with the HIV, it will produce a large number of the serum antibody to the epitope of these viral proteins. Now, these serum antibodies to the HIV can be detected with the help of the indirect ELISA within the 6 weeks of infection. So what they do is, if you see, the wells are already antigen coated over there. Now, as the person sample is being loaded onto these wells, what happens is, if the patient sample are containing the antibodies, they will go and bind to the antigens over there. As the binding takes place, there will be a secondary antigen which will be added or loaded onto these particular well, which contains certain enzymes which can convert the colorless substrate into the colored product over there. So then the coloration will come into the picture. The next one is the next form of the ELISA, which is called as the sandwich ELISA. As its name is suggesting, it means that the antigen is sandwiched between two antibodies over there. Now, the antigens are generally detected by this particular method, which is called as the sandwich ELISA, and it is very commonly used in detecting the malarial antigens in the blood samples. Now, in this particular technique, what is being done is, the antibody which is specific to a particular antigen, they are coated onto the well. Then the antigen are being added. So these antigen, it comes and reacts and binds to the antibodies over there, and as this binding takes place, what happens is, secondary antibodies are then added. Now, these secondary antibodies have got the enzyme which is linked onto it. They get bound to this one. So we can see over here, there is a sandwich which is formed, in which there is an antibody, then there is a antigen coat, then there is another antibody which is on the top of it, and then subsequently the substrates are being added. So in the presence of these enzymes, these substrates get converted to colored product and the coloration comes up. So we can easily mark that this particular well contains this sample in which the patient is suffering from malarial over there, malarial infection. So these particular sandwich ELISA gave a very important medical diagnostic kits also. Now, the next important thing which was developed with the help of these antigen-antibody interaction pattern was the Western blotting. Now, this Western blotting, as we know, it is one of the blotting technique along with the southern and the northern Western blotting technique. What happens is, in the Western blotting, we identify specific protein in the complex mixture of the protein which are accompanying in that one. So this particular technique was called as the Western blotting. Now, this Western blotting technique became a very interesting technique for confirmatory testing for various antigen or the proteins, like in the case of the HIV, one of the, or the viral proteins also, it can be easily utilized. So the Western blotting technique became very instrumental, and it again used the antigen-antibody binding pattern, which is very specific. So in this one, what happens is, the gel is prepared, which is the SDS PAGE gel which is prepared. So sodium dodecyl sulfate PAGE gel is prepared, and then it is electrophoresis. So all these proteins have been negatively charged, so it moves towards the positively charged, and depending upon their sizes, they get segregated in those particular wells. Then this particular gel proteins are being transferred onto the nitrocellulose membrane with the help of the blotting technique. Subsequently, what happens is these nitrocellulose membranes are being added with certain antibody which are specific to a particular antigen, and it is being rolled over there over a period of time. Now, as we know that these antibody will be very specific to a particular antigen or the epitope which is present in that particular plot, it will only go and bind to that particular plot over there. So we can see over here at the bottom there is an image of a protein which is shown, and there is a mark radio labeled protein because these antibody are being radio labeled, so it can be checked with the help of the autoradiography method over there. So we can see over here the protein, there is a presence of a dark label over there, and this dark label gives us a indication that that particular protein is being present over there. So this antigen antibody interaction played a very important role in the detection of the specific proteins over here, and this Western blotting technique has now become one of the most significant and instrumental in separating and detecting the proteins over here. So with this note, I come to the end of my lecture. So in this part of the lecture, we discussed about few of the important techniques or the medical diagnostic tools and the immunological tools which have been utilized and developed over a period of time based on the application of the antigen antibody interactions over there. So we'll take up this lectures further in the next part of the lectures. Thank you very much.